All right, guys, we'll go ahead and get going today. Appreciate you coming and listening to what Dr. Campbell has to say today out of Penn State University, and Dr. Addie Waxman will give a few presentations afterwards as well too on some of the studies and field stuff we've been working on with 1,4 DMN. So we appreciate you guys coming and showing up today. And first off is Dr. Campbell out of Penn State University. If you wouldn't mind giving a quick update to everybody here, what you do and what you've been doing the last 3 or 4 years with us, and then we'll let you take it away.
Okay, great.
Thank you, Mike.
All right, I'm a professor at Penn State. I work on plant molecular biology, so I do genetics. And I'm interested in the genetic effects of post-harvest physiology. And the 1,4 group developing DMN as a sprout regulator was very interesting to me. I've spent many decades working on sprout regulation, not just in potatoes, but in perennial plants. And I'm interested in how DMN functions. And that's what I have been spending the last few years working on, is genetic aspects of how DMN works. I live in a very different world than you do. You're very applied, and my work tends to be focused very detailed, and I'm zeroing in on what potatoes are doing and how an individual potato responds to DMN to try to figure out what's going on. That said, our projects have expanded a little bit, and I'll explain that as my talk progresses, to not just looking at potatoes anymore, but other things that are sitting in the bin with potatoes. So let me get started, and what I'll first talk about is how DMN affects potatoes and how potatoes respond. Now, potatoes respond because there's genetic responses. There are genes that get turned on in response to something, and there are genes that get turned off in response to something. Take heat, for example. If you were to take a plant and heat stress it, it gets shocked by the heat. It turns on genes to try to protect itself from the heat. It turns off genes that are unnecessary. The same thing happens in DMN. You apply DMN, and the tuber is turning genes on in response to DMN, and it's turning genes off in response to DMN. And that's what we were looking at for the last couple of years. Now what that gives us is gives us some idea of what's going on in the potato, and that what other things might be happening that could be of use to processors and growers as far as application of DMN goes. So let's take a look and see what we got. Over the years, we looked at a number of things as far as sprout regulation, not just DMN. We looked at time. What happens to a potato just when you harvest it and it just sits there, and you put it in a storage shed and just let it sit for 6 months. We looked at that. We wanted to see what happened there. We also looked at bromoethane, which is a compound that breaks dormancy. It's a very strong compound that actually we can dig something up like a russet Burbank out of the ground 2 weeks later, treat it with bromoethane and get it to start sprouting. I wouldn't recommend it. It's pretty nasty stuff. And then another one is 1-ethylbenzyl-3 nitroguanidine, which is another compound that induces sprouting. We call that NG for short, just because I couldn't stand that name. And then we also looked at sprout suppression, what turns sprouting off. And we looked at gene responses to CIPC, which I won't talk about today, but I will focus on gene responses for DMN. When I say gene responses, I'm going to give you some basic biology here. DNA is all the genes that are sitting in a cell, sit in DNA, but they don't do anything. DNA is like a hard drive, and it doesn't do anything. It has to get copied. Once it gets copied, then that gene starts to become expressed. So what we did in our experiments is we looked at RNA levels, which are the copying from the DNA. Once the copies start to be initiated, we know gene expression is changing. Sometimes the copies get turned off. So we can measure both genes getting turned on and turned off. Okay, and a lot of this is happening in the last few years because the technology is here now. We couldn't do this 20 years ago. This was not possible. So now that we have these tools, we can do a lot of things that we could never do. So it became really interesting from us from that perspective. Okay, so I don't live in your world. I live in a world where I'm looking at only a few tubers at a time. I don't want to have complexity of a giant bin. And the problem with a giant bin is, are you sure that tuber over there and this tuber over here got the same dose of DMN? I need to make sure that when you say there's a dose of DMN, everybody gets the same dose. Because I've got to have control. So I have these little bins. And these little bins, I actually apply the DMN using a vapor. And I've got a filter paper that sits on the top of the bins, and I can put DMN. Now, in here, the tubers are cut. We also did this weird, crazy experiment. I don't want to talk about it right now, but we took a tuber, cut it in half got treated with DMN, half got treated with control. What I'm going to talk about today is everything is just a whole tuber treated with DMN. So this is another experiment I'd love to talk to you about if you want to hear about it. Okay, so I have these small chambers. I put the tubers on the bottom, only as one layer. We treat with DMN. We take residue levels by sending it to dichlor. Dichlor tells us how much DMN is on the surface. So we sort of have an idea of what we're dealing with. Okay. We get some fairly good response of sprout control, depending on the tubers. We used a number of different tubers. I think the last set of tubers we used were the chipper. I'm sorry, I live on the east side of the United States. It's really hard for me to get a good russet. They want to grow russets in Pennsylvania, but let me tell you, it's pretty sad. But anyway, so most of these are going to be the chipper, I think, in this next set of experiments. We're also concerned that a tuber is a very complicated structure. You know, you think of a potato, you think, well, there's a potato. No. There's a sprout, there's paraderm, there's inside where the starch is. So we wanted to say, if we're looking at sprout control, we only want to look at the sprouts. So we treat with DMN, and we use this instrument here that's called a microcurette. It's a little tiny metal cup. It's actually used to do bioassays on people. But we can actually use this to pull out the meristem from or pull the eyes out, and then we can isolate the eyes and then look at gene expression and adjust the eyes. So what we would do is treat with DMN and then pull out these little meristems and then look at gene expression there. Because I don't want to know what's going on in the rest of the tuber. It's just going to blow my mind. It's too complicated for me. I like to keep things simple. We did a number of things. One thing that we were interested in, and I'm sorry, it seems to have changed. That's fall. We did three different treatments. One where we treated with DMN in the fall. That's pretty close to harvest. So it's about 2 weeks after harvest we treat with DMN. We put them in storage. We also had a treatment which we would call late winter or early winter. In our case, for the chipper, that was about the beginning of January. A little bit later, actually. A little bit. I can look at the dates. I think it's towards the end of January, actually. And then we did another one later in what we call spring, and that would be late March, early April. The difference there is that there's a physiological difference in the tubers. When you first take a tuber up, it's dormant. It's not going to sprout. If you treat just a few weeks after harvest, it's not going to sprout, and you've treated with DMN. So we want to know, what is the DMN response here? We'd also like to know, what is the DMN response here? After it comes out of this endodormant state, or the state where it's not going to sprout, and you put it in storage, and it's sitting in storage, certain varieties, russets are a little bit longer dormancy, but a lot of varieties, if you pull them out of storage and bring them back to room temperature, they'll start to peep. And as the meristems start to peep, you know that the meristems are active and they're no longer asleep. So we wanted to treat them also at that point where they've lost this endodormant state. And then we also wanted to go way out when we've got them in storage that we know that if we take them out, they're just going to go crazy and sprout like mad, right? So now we've got 3 different DMN treatments as we progress through the year. All right, the DMN was treated for 2 harvest years, so we did this 2 years in a row. So we harvested in the fall, did our 3 treatments across the harvest time, the storage time, and then we did it again the following year. And we did that so it was a little lighter one year than it is the next. It's a little bit warmer, you know, so the harvests are slightly different. So we want to try to mitigate for that. Again, we're going to use the chipper. The tubers that I have were provided by Troyer Farms. I grow my own tubers. I actually contract out to Troyer. Troyer was actually growing them for seed potatoes for Florida. So they would harvest and then call me up and then say, we're going to harvest and send them off to Florida. They were using them for cable. So we would be able to just go in there and load up our cars with tubers. And then we did a 3rd-year harvest where we did a different technique to sort of duplicate this, to just double check it on the individual genes. And I'm not going to talk about that. Okay. So what the heck does this mean? I I love to show this slide just because to me, this sort of shows you what's going on with the technology. Potato has roughly about 25 to 30,000 genes in it. Those genes come in different forms. They have different structures to them. And when we look at this kind of experiment, we will isolate this RNA compound, and then we'll sequence it, and we'll map it back to the genome and say, where in the potato genome is this gene turning on, turning off? So it gives us a lot of other complexity. But what I need to explain to you, though, is that we're looking at 41,000 different RNAs that change in the first year. And then we're looking at 36,000 RNAs that change in the second year. That's huge. That's all these different genes in response to the storage process. What we can tell you is that about 17% of the genome, or all the genes in potato, respond to DMN. And the second year is about 18%. So it's between 17 and 18% of all the genes in the potato respond to DMN. Now, only in that meristem, though, right? So we're only looking at that meristem. So that tells you that potatoes respond to DMN at a fairly high level. 20% is pretty high. So when you get all these genes, you can say, oh, so what? He's got thousands of genes that respond to DMN. Well, what we could do is we can look at these genes and say, well, what do they do? And that's where the technology has changed. About 2011, the Potato Consortium got together and they sequenced the potato genome. And they sequenced the genome and they looked at all the genes and they looked at how they're sequenced and they tried to figure out what they do. And what we did is we said, can we have your files? Can we then take your files and say, well, what do we see compared to what you tell us is in the potato? And we break them down by what they do. So the number of genes that are responding to DMN, we could describe what they do. And here we can say that some of them are metabolic processes associated with carbon processing. So they're the carbon metabolism of the potato. Some associated with cell wall. In this case, this is lignin biosynthesis. These are things repressed by DMN. So think of this. DMN treatment is turning things down. All right? So these are repressed genes. Why are these genes going down? Well, it makes sense. If you think about it, there's a handful of genes associated with cell wall production. They get turned off. Well, that makes sense. DMN is suppressing growth, right? It's suppressing sprouting. It's suppressing cell division. Why do you need cell walls? So the cells are turning off the ability to make cell walls in response to DMN. There's also protein catabolic processes. I don't want to get into that. That's essentially catabolism is breaking down, so protein breaking down of proteins. One is response to stress. We have some genes that go down in response to stress. And then you'll see on another slide some genes go up, so it's complicated. Sterol biosynthesis, sterols go down. Auxin biosynthetic processes. Auxin is a hormone that's associated with the growth of the tips of shoots. It does some other things, but auxin biosynthesis, that's why Christmas tree has that nice pointed shape. The shoot at the top is inhibiting auxin biosynthesis in the other buds. So what we're seeing here is that these are things that are associated with auxin signaling, and they go down. That makes sense to us. And phenylalanine biosynthesis, don't want to go in there. And then here one for meristem function. This is the actual meristem. There's a gene that's associated with the function of the meristem. That goes down too. Okay, so that sort of makes sense. These are genes that are upregulated by DMN. Now, these are upregulated both in endodormant state, the dormant state, so whether it's right after harvest, whether it's in the winter, whether it's in the spring, and it happened in both years, and this is what they do. There's a lot of them. Okay? The ones that I want to sort of point out to you, though, are right here. This one is really interesting. This one's called Win2. It's a wound-inducible protein. So in other words, it's a wound-inducible protein that's turning on in response to DMN. It's a stress response protein. It actually is responding to DMN as a stress-inducing compound. Okay? Another one that's really of interest to us, and this one showed up a lot, is osmotin. And there's a number of osmotins that are on here, and I don't want to go through it. But that protein is induced by DMN. And what osmotins do is they regulate water. They regulate water flow between cells. That's a gene that's being induced by DMN. Okay? So why that is happening, we're not quite sure, but it does tell us that DMN is inducing stress, and it's inducing stress associated with water stress. So it's almost as if you're telling the potato, you have an issue with water flow within your cells. And then DMN is essentially inducing genes to try to mitigate for that process. Okay? There's a bunch of other things in here that are really quite interesting, but that more for long stories. But in summary, DMN decreases gene expression. And it does that in some genes. And those are genes, a lot of them are associated with activity of growth. Okay? Turning them down. DMN causes some genes to increase in activity. A number of those genes are responses to stress. There are also some genes associated with bacterial response. So in other words, if I went back to that sheet, you would see there's some response to bacterial stress. So, you know, if you take a bacteria, these are experiments that people do in other plants. They'll take bacteria, they'll grind them up and kill them. And they'll just take the bacterial extract and spray it on the plant and say, well, what do you do? The plant goes crazy and says, oh, I see bacteria. And we're seeing the same kind of things. We see some of those genes get induced by DMN. So now I think that's an overall stress response. DMN's saying there's stress. Some of that stress is a response to bacteria. Non-dormant tubers have a greater response to DMN compared to dormant tubers. Since we did a treatment where we took DMN and treated right after harvest, a couple weeks after harvest, we also had tubers that were in storage that we treated later in the year. Again, we're actually treating them again. So we treat them after dormancy is broken, but they're still in storage. The tubers that are in storage, where they're no longer dormant, have a much greater response to DMN. So what that tells you that the response to DMN is not the same depending on how long the tuber's been in storage. As the tuber ages and sits in storage, its response is stronger to DMN.
Mike?
Yeah.
They still have a response in the fall.
Yes.
But
yes.
The response is just greater.
It's much greater. Yeah, it's like a rheostat in the room, right? You turn the lights on, you turn it on and they're dim. You turn them on really high and you turn them on, they're really bright. So the response to DMN is just much less. Now, in a way, it kind of makes sense because the tumors are dormant. They're not doing anything. They're just sitting there. There's no metabolic activity. As you get out of that dormancy, there's an ability to have metabolic activity. So DMN is going to shut that down. Okay? I'm not saying there's no response to DMN when they're harvested. There's still a response. It's just much greater later. Okay. So what I can tell you about DMN is that it seems to be inducing a stress response. The response to DMN seems to be a much greater response as the tubers become more active. That, in a way, that kind of makes sense because DMN suppresses growth, seems to be inducing a stress response, and also it seems to be inducing a stress response associated with drought or water stress. Now, while we're doing these experiments, I have a number of students that work for me. And one of the students came up to me and he said, you know, when you put DMN on potatoes, it's not just potatoes that are there. Potatoes sit and they're covered with all sorts of stuff, right? They come out of the ground and they're just covered with stuff. What happens to that stuff when it's treated with DMN? And I told him, I said, I don't have a clue. I tell you what, since you brought it up, I've got a project for you. I said, I want you to go and get some baker's yeast. Yeast is an easy thing to grow. And I want you to grow it in the lab, and I want you to treat it with DMN. And spike with DMN to a level that's fairly close to what you would see, that a potato would see. So he had this culture, and this is the same yeast you used to make beer. So he was like, yeah, I'm going to grow this. So he's growing this big vat of this yeast, and he spikes with DMN, and he's doing cell counts. And since he brought it up and he was willing to do the work, I said, I want you to do cell counts every 4 hours. So he came in the middle of the night, and he took samples from his cultures and put it under the microscope and did cell counts. So we got cell counts, and what's happening to these cultures after we hit them with DMN? Now, normally, when you add a culture just takes off and it goes into this thing called log phase, where it just grows like crazy. When we hit it with DMN, it stopped. The culture stopped. He took the cell counts, he said the cells There's so many cells per mil of culture, and 2 hours later it's the same number of cells, and 4 hours later it's the same number of cells, and this went on for about 7 days. And then after 7 days, the cultures just took off and went going again. And that just puzzled me. You add a little bit of DMN, and we're not talking a lot of DMN. We're talking 7 microliters of DMN to 500 mils of culture. Okay, so I got this big thing of culture, and I add just 7 microliters of DMN, and the culture just stopped. And it sat there for a week. Well, 7 to 10 days. And then, poof, just recovered like nothing ever happened. That project sort of sat on the wall for a while, and we didn't, you know, put it on the shelf, and we didn't really work with it for a bit. And then in biology, they've got this new system of looking at what they call the microbiome. They can do this where they can look at all the surface of your skin and look at all the bacteria and all the fungi that live on the surface of your skin and see what's there, right? And I thought, you know, I wish we could do that with potato. Maybe we can even do that and actually start to look at the community that lives in a bin. Not just potatoes, but everything else that lives in the bin. So we started those experiments, and we started this thing that we call the microbiome, where we looked at bacterial communities and fungal communities. And that's where the tubers were cut in half and all sort of. But to keep that simpler, what we also started to do was saying, well, all right, what if we found certain bacteria or fungi that are living on the surface of the potato, cultured them, and then treated the cultures with DMN like we did the bread yeast to see what happens. So in that case, we decided to go for specific plant pathogens. Could we find specific plant pathogens on potato, and then say, well, what does the plant pathogen do when it sees DMN? So what we did is we started with fusarium. And the reason why that happened is because my son was actually working in a lab at another school working on fusarium. And we were having a conversation. I said, I'd really like to work with some plant pathogens, but it's a real pain in the neck to get them because you can't just ship plant pathogens around. There's a lot of regulations and stuff. And he goes, you should just get your own. And I said, well, how can I do that? And he goes, here's a recipe. So he gave me a recipe to isolate fusarium off the surface of a potato. So I told the student, all right, what I want you to do is I want to take a potato, scrape the surface of the chipper potatoes, put it on this media that selects for fusarium, and get me some fusarium cultures. And the student was Rachel Adams, and Rachel did a very nice job, and this is putative fusarium cultures. There's some other things on here besides fusarium, and but this is a special selected media to get fusarium. Fusarium is a fungus, so you've got these fuzzy things that are growing there, and they're fusarium. And I said to Rachel, how do we know they're fusarium? What we need to do is we need to DNA fingerprint them. I'm a molecular biologist. I like to do this. I sequence DNA. I can sequence DNA from these cultures and then compare it to other people that work with fusarium, and they'll tell me what this is. So we did that, and what we found is a fusarium strain that we isolated from potato, and I'll show you some pictures of the fusarium strain, specific fusarium strain in a minute. And then we identified that strain using DNA sequencing. We also isolated spores from that, and we took the treated spores and we germinated them in cultures by spiking them with different amounts of DMN. Because we want to know, well, what does DMN do, not just to the fungus growing, but how does it do in response to, how do the spores germinate in response to DMN? Does DMN kill the spores, prevent them from growing? What does it do? So what we were looking at in that case, we were looking at the fungus growing on a plate, and we're also looking at the spores we isolated. Do we kill them and keep them from germinating and growing at all? That would be interesting to know. Does DMN kill fungi on the surface of the potato? But after we did that, though, it turns out that when we treated the spores with DMN, there was some time that they would sit on the plate, but then they would recover and germinate. So in other words, when we treat with DMN, we isolate fusarium, we make it spore, we collect the spores, we treat the spores with DMN, put them on a plate. The ones where we don't treat with DMN, the spores germinate right away. The ones that are treated with DMN, wait a little bit, and then they germinate. So the DMN did not kill the spores. So it's not killing the spores on the surface of the potato. When we grow cultures, and we look at the culture growing on the surface of a plate, it slows the culture growth. So the culture, and I'll show you a picture in a minute, the cultures grow on the surface. If we treat with DMN, the culture slows and slows down and doesn't grow as fast. And it'll eventually recover again. So this is a picture that Rachel came up with, and I wish you wouldn't just use your cell phone and hold it up to the window by the lab. I thought she would be a little more, but that's Rachel. But that's okay. So what this is a fusarium culture that's isolated. We know it's fusarium. We sequenced it. We identified as Fusarium oxysporum. We grow that out. We can germinate the spores on a plate. We can get them to respond. And then when we grow them and get the cultures to start to grow and then put them in with DMN for 2 days, this is what happens through a control. This is what happens when you're exposed to DMN. Now, when I say DMN treated with 2 days, we're putting them in the same container we would put them in with our potatoes. Okay, so the DMN misted into our control chambers, and the cultures are fine. Now, the cultures are slow to growth. If I take those out and keep them away from DMN and let them sit on a shelf away from DMN, they'll just recover and look like that. So what that's telling us is DMN is suppressing the growth of the fungi, but it's not killing the fungi. So that's where we come up with this term fungistatic. In other words, it's slowing and preventing the growth of fungi while it's present, but it's not killing the fungi. Yeah?
When you take them away from the presence of DMN, do they start growing pretty quick?
Yeah.
Or is there a little bit of a,
Well, the problem there is that for fusarium, yes, they grow pretty quick. Some other fungi is a little bit slower. So different fungi that we looked at, the response to recover from DMN is different depending on the strain. Fusarium is pretty wicked and grows really fast. So look how fast those grow across the plate. So these will recover probably in a day or two. All right. I have some other strains I'll show you later that took a little bit longer to recover, but fusarium tends to go pretty quick. Okay. So this is a strain of fusarium associated with dry rot for potato and tomato, and also is a problem with peppers too. We then started to look at some other fungi that we could get a hold of. We could get some plant pathogens shipped to us. And this is one that we actually purchased from a company called American Type Culture Collection that's in Maryland. And this is Aspergillus niger. And niger causes black rot on onions, rot on tomatoes, tuber rot on yams. This is not a big problem for potatoes. You probably don't see this as a big pathogen problem in potatoes. But we were interested in what does Aspergillus niger do in response to DMN. And the same thing, if we put the Aspergillus niger in a control chamber with no DMN, we get our cultures to grow like that. Now, yes, the morphology is very different than fusarium, so it just is. And then if we treat with DMN, we get something like this. It's really hard to see in this picture, but there are colonies there. There are cultures there. They're just not big like this. And that's after our 2-day exposure to DMN. So that's what we see. And these will eventually look like that if we just take them away from the DMN and let them go. What that time frame is, I don't know. We didn't do it for each species. This is alternata. Causes early blight. Here it is on our control. Here is our DMN plate. You can see there's little tiny dots there, so it's still there. It's still present on the plates. They all go in at pretty much that stage. So this was put into the chambers at pretty much that stage. And this is what happens after 2 days, and that's what happens in 2 days of DMN. So it just sits there. We can get it to take off and start to grow again someday, but we pull it away from DMN. This is Epicoccum nigrum. This is a plant pathogen on many different species. And again, here's the colonies for it on the control plate. Here it is on DMN. Again, there's some tiny little colonies on there that you probably can't see unless you come up and stare at the slide pretty close. This is an interesting one. There's a number of people that work in my lab, and one student works with American chestnut. And she was looking at isolating American chestnut blight. We have these hybrid chestnut trees that had cankers on them. And everyone was looking at the cankers, and nobody thought they were chestnut blight. We didn't know what they were. So the student isolated a fungus from those cankers. And we sequenced it to figure out what it was using DNA sequencing. And it turned out to be this species here, Pneumoniopsis smithglivii. And this has been reported to cause cankers in Europe and on chestnut trees in Australia, but it's not been known to cause cankers in the United States. the student isolated this culture, and we thought, what the heck? Let's see what happens when we put pneumoniopsis and expose it to DMN. And we see the same thing. The culture grows very sloppy. It's a weird network of fungus that grows across the surface. We can never really get it to be nice and tight, like you saw with the aspergillus. But it essentially grew as a fairly robust culture in control, and it's definitely wimpy when it's exposed to DMN. This was very interesting. She presented this in an American Chestnut Society meeting, and all of a sudden everybody said, yeah, we get these weird cankers too, and we were always wondering what they were. So the student was all happy that she was the first one to sort of report this in the United States. But it's also inhibited by DMN. I don't know where that goes. But, this is Pestaloleopsis. This causes trunk disease. This student working on chestnut also found this species. And again, it's the same thing. Here, the cultures are not very nice and bright, but you can see there's colonies here. They're fairly big, and we don't see them on here. That little square there is actually a piece of the Petri plate. There's a little stamp on the Petri plate, so you can put writing on it or something. I don't know. But the same thing. It's inhibited by DMN. And then this is FOMA. FOMA also shows a nice response to DMN. Nice, beautiful, clean colonies on the control. Very tiny. You can see the tiny little dots here in the presence of DMN. It causes a stem rot and leaf rot in multiple species, not just restricted to potato. And this is Pythium. And Pythium, again, is a sloppy kind of culture when it grows. Causes shell rot or leak. It just is robust when it grows without DMN. With DMN, it's slowed down quite a bit. So what that sort of tells us is that we've got a whole bunch of different fungal cultures, different kinds of fungal species, and they all seem to be inhibited by DMN, by the presence of DMN. When DMN is there, they slow down the growth. Very similar to potato.
Mike?
Yeah.
We had one question.
Yeah.
Can you slow a fusarium down enough that the potato's defenses can take over at that point, or does it affect the defense?
I'll tell you what, I do the basic stuff. Someone's going to have to do the applied stuff to find out if that's true. The smoking gun says yes. That's all I got. I got a smoking gun, right? I can inhibit fungal growth. If you give me the fungal cultures, I can inhibit their growth by putting them in a chamber with DMN. Whether that causes a viable application of DMN to bring a lot of money in someone's pocket, I can't say that for certain because I never did the experiment. Okay? This is my job. So, okay, this is another thing that we were thinking about. You know, those fungal cultures are growing really nicely, but fungi are very complicated too. They have developmental stages, and they go through different developmental stages. I had one student who was growing Aspergillus niger, and she was growing Aspergillus niger so the niger cultures would grow, and she can get them to induce a lot of spores. And we wanted to know, well, what happens if you're getting to that developmental stage where you've got a culture that would sporulate and make spores, but you can put it into the presence of DMN. So what she did is she took a Aspergillus culture, and in the control situation, this is a very mature culture, so it's a big disc of fungi that she puts on the plate. And the disc is about a centimeter across. And that culture she puts on a plate, and that culture is so mature that those fungi are ready to form spores. All right? The spores are how the fungi replicates, right, and sends itself around. So what she did is she put one in the control plate, put one in the DMN, and what DMN seemed to do is prevent sporulation. So it prevented the Aspergillus from forming spores. And we did it at different doses. I don't want to get over what that means as far as our concentration of DMN per liter. But as we drop it down, we still see the same response. It was a dose response. So as we go up, it didn't seem to make any difference at this dose or that dose. So it prevented sporulation. Which gets to the question that maybe you can prevent spore growth, but also maybe you can prevent it from spreading within the bin. You've got a hot spot in the bin where something is taking over like crazy, it's going to spread, right? That might be able to slow down and spread. Again, smoking gun, I didn't do the experiment. So. So what do I know about this? DMN and fungal cultures. DMN slows the growth of multiple fungal cultures. We've got a bunch of different fungal cultures with the same response. Put them in a chamber, mist in DMN, slows the growth. It decreased sporulation of aspergillus. So in other words, aspergillus didn't form spores. I don't know if it does that for every species. Some of these species, it's impossible to get them to sporulate in culture. You know, it's difficult to grow them. And that's an experiment that would be interesting to try. But it did decrease that. But that said, though, I can kill a fungal culture with DMN, right? I can do that. But I can kill a fungal culture with water, right? I can kill a potato with water. So it's a question of dose. At the dose that we use in bins, and the dose that you normally see as an applicator, DMN is not killing fungi. It's slowing their growth. So DMN, if it's doing anything, it's fungistatic. It's slowing down the process of infection. Now, the question that you had about whether you could get the potato to come up with defense response, if you go back to the gene expression studies, and you look at the genes that are induced by DMN, a lot of them are stress-induced genes. And some of those genes are stress in response to bacterial infection. So DMN is inducing stress response. As it induces stress response, is it inducing stress response to increase protection against pathogens? Those genes are there, they're induced by DMN. So the question then is, even if you did this experiment, where you said, I'm going to take a bin, and I'm going to treat it with a pathogen, I'm going to close it up, and I'm going to treat it with DMN, I still don't know. I'm still not convinced that you can determine whether the prevention of a fungal culture within a bin is because the DMN is inhibiting the fungus, or the DMN is inducing the potatoes to become more resistant to the fungus. Maybe it's a little bit of both. It's slowing down the fungus, and it's inducing the DMN, the potatoes to induce genes to protect itself against pathogens. So there's sort of that double edge there that could be there. I'm sort of hoping that Addie's going to come up here and tell us something about what happens when we put DMN in a bin with pathogens and what happens to storage. Those are experiments that I don't have, because I live in the basic world, right? I don't do all the flat stuff. Okay. Oh. We're frozen.
Is that the last one, Mike?
Yeah, I think it's the last slide anyway.
What about, are you digging around with bacteria, and you feel like you're seeing, if you have,
For bacteria?
Yeah, you feel like you're seeing some of the same effects?
Yeah, I didn't want to talk about that because those experiments are still going on. They're still kind of messy. I have a student that's working with, actually, he didn't want to work with bacteria that infect potato. He wants to be a doctor. So he wants to work with streptococcus. So we're working with streptococcus. So he's working with, actually, he's working with a MRSA-resistant line too. MRSA-resistant streptococcus. He's got it in the lab and grows it. And he's treating with DMN. And it does inhibit its growth. We're still trying to figure out the dose. It's a little bit challenging to do that because yeast I can grow in a big barrel and treat with DMN. And the amount of DMN you need to add is very small. I do not want him growing a MRSA culture in my lab in a vat that's like this, okay? Just not going to happen, okay? So he's got to come up with experiments to dilute the DMN. And it's really hard to dilute DMN. You can't put it in water, right? You guys work with this stuff. So he's got to put it into something like DMSO, and then he's got to spike the cultures. But then he's adding DMSO, so he's got to do the controls. So he's got to treat the controls at the same amount of DMSO. So the experiments have been kind of complicated. Yes, it does inhibit streptococcus growth. How much and what dose, I can tell you yet. We're still working that out. But what he did is he did dose response. So what he did is he took a culture and he dosed it with so much DMN and it killed everything. I mean, he had so much DMN. Yeah, what? Right, but I could have done that with water, right? I don't know where that window is. We're looking at what we call the LD50, which is a lethal dose of 50% of the culture. So we're almost there. It comes out to be about 200 micromolar. Oh, it's 150 micromolar. I don't know if that knows anything. Okay. What else?
Did you, like the Petri dishes, you just applied DMN to it and pulled them out? Or
No,
they had...
we tried that. The problem then is when you pour a Petri dish, you're pouring a Petri dish of hot agar. And we had students trying to put the DMN in the agar. But DMN is kind of volatile, so you put it in hot agar and it just evaporates out. So what we did is we put the cultures, the Petri dishes, we put the fungal cultures on the Petri dish, started the culture, and then put the dish in the chamber and then mixed it in the DMN. It was just too complicated to try to put it in the culture with the fungus. Because it's in agar. Agar's hot when you pour it, so. And DMN won't.
So those Petri dishes stayed in an environment that was...
Same with our potatoes. It's like you have your bin, we put a culture of a Petri dish with fungal culture in it, and then apply the DMN. We thought that was most realistic, right? Because that's something that you guys are going to see when you apply. So, yeah.
So back at the beginning, you talked about, you did a potato, the DMN. So obviously you cleaned that potato to remove the dirt and organisms off the potato?
No, we don't wash them.
You didn't?
We don't wash them. No.
Because then the second point you brought up was you actually treated some dirt pathogens in the dirt. So there was technically some on the potato.
Oh, yeah, there's some dirt on the surface. There is stuff on the surface.
All right.
Your growers aren't washing their potatoes before
No.
they go in the bins, and I didn't want to do that because that becomes a real interesting challenge because you got some years, if you're harvesting in a wet year, you're coming in with a different microbiome than you would on another year. And that's a whole different story, right? That's a nightmare. I've had tubers I brought in that are just caked in mud. And then other, you folks in Idaho, it all falls off because it's all sand. But where I am, it's a little more challenging. I think it's a little more clay in our soil. So. But you're right, that it's complicated. But I still think, as people that store potatoes, right? There's more going on there than just potatoes, right? You've got this bin of potatoes, and you've got everything that's living on the surface of them too. I think there's other potential. There's potential there. Now I think you do treat with peroxide, right? And that's an idea to try to control pathogens. But there's more that can be done. And whether DMN's doing something also is a great question. So, yeah.
Did you come up with any theories on how it gets in the cell or how it leaves out of the cell?
Well, it's a very hydrophobic compound, so it's not water soluble. So it's going into the membranes. So it's going into the cells are surrounded by a lipid membrane. The DMN, I think, is going into the lipid membrane of the cells. It's not going very deep into the tuber as far as we can tell. When we did the microbiome experiments I didn't talk about, what those are is you isolate everything on the surface and just sequence everything on the surface and then figure out what species you've got. Well, I didn't show you those experiments. When we did those experiments, we took a tuber and cut it in half. And then we took one half and put it in the control, one half we put into DMN. And we did that because we like to have a wounded surface, right? We've got a half of a tuber, that's a wounded surface. And then we treated with DMN. And then when we did the residue levels and said, where's the residue level? The residue level was much higher on the uncut surface than on the cut surface, right? Because the cut surface is very wet, and DMN does not like water. And it's not going to stick to the wet surface as well as it would to the other surfaces. So I think it's going into the cells through the membrane, and it's not going through as in the aqueous part of the cell. That said, why are genes associated with water regulation being turned on in response to DMN? And I think that's because a lot of things that control water regulation sit in the membrane. So if DMN's penetrating into the membrane, the cells are saying, well, my communication to the outside world is getting screwed up somehow. Something's happening here. And that's affecting water transport through that membrane. So that's a theory, right? I don't have the real detail to show the physiology of that. What else?
Any other questions for Dr. Campbell?
What do you see when you look? I don't live in the world where I see bins treated with DMN. Do you see bins when they're treated with DMN coming up with a response to, say, pathogens that are different than you do in a bin that might not be treated with DMN? Or do you don't know?
2 years in a row, I treated Umatillas for growers that every year they have fusarium issues.
Yeah.
These last 2 years in a row, the only bin they had fusarium was the one they did in DMN.
Yeah. Makes sense. It makes sense.
Which is an early application too, right, Brian? Right?
I was there 24 hours, yep.
Yeah.
Early on.
24 hours.
Right.
going in.
You see that? All right. Now you get to the point where, well, why you apply DMN when you first harvest is they're not responding as DMN to a very strong response, but maybe you want to apply DMN because if you've got any kind of pathogen issues, that's one thing you're doing is you're also shutting down the pathogens. And you're not just preventing sprouting, but you're shutting down the pathogens too.
And I got one storage, it's 300 feet long, 220 feet of potatoes were in there. Simplot told them, McCain's wanted them to switch sheds to go digging somewhere else. So they left that field, so 220 feet out of DMN within 24 hours. 5 days later, they put the rest of the 80 feet in the bin, and I didn't DMN it. I could literally wear a GoPro and walk along the catwalk, and you see cut like a knife, all the rotting potatoes down in what's not treated, same field, sitting 5 days later. Like it's
Another thing I was wondering about, when you treat with DMN, do you see a difference in shrink? Or do you see a difference in water loss? Because it looks to me when I look at gene expression, I'm seeing a lot of genes associated with water stress. It's as if the tubers are saying, I'm water stressed, I gotta do something about it. Which means that controlled tubers are not responding that way. And I don't know. I
80% of my growers don't humidify.
yeah.
No humidifying at all. Right now they're saying they can't believe how tall the piles are staying up where they filled them in. Before they dropped 3 feet, the pile like,
Without the
yep. It's huge this year in my area. Guys, and we got issues.
So other
Good
growers-
potatoes, they don't humidify.
That makes sense too. It all sort of fits what I see. So,
Yeah, Dr. Campbell, I'll speak in place of my applicator that I deal with in Michigan. And he's seen this disease issue, the effect with DMN for a number of years. It's probably been 6, 7 years or so now, this time. But it actually, I was at one of the meetings, and it actually came from a grower that came up to me and said, our product does something for rot issues. And now I know that there are growers out there that are using this religiously for that program. I visited with one grower where, he says, I know something is going on with this. My storages are better year in and year out. But you can tell that there was a little bit of doubt always, whether there's something going on. But this really, I think, substantiates or proves that there is something going on.
well, it's what I see. That's what I think would happen, and that's why I'm asking you. Is that what you see?
Yeah, I mean,
because that's what I think should happen.
Right.
But I don't deal with big storages. I deal with potatoes on a single layer and a treatment. I know where I got the same dose. Another thing is, DMN mixed with CIPC. Is there anyone who ever mixes it?
Yep.
What's that response look like?
I like that. That's an excellent way to treat it. I think I just did some last week, but it works really well.
Yeah.
I've seen sprout controls, I really like to treat it.
Right.
Yeah. This year I'm really seeing them when we saw in frost damage. I treated a lot of 25 to 30% in frost damage in bins on door closure with DMN. And they're still storing the bins now. And
Really? Prevented
frost never took off.
Frost damage is what's happened.
Just
There's a
hold
lot
it,
of
yeah.
cells break open because the ice crystals, and they lose water, and then that water is just a magnet for pathogens. So,
How late do they plan to store some of these bins?
All those guys are now shipping to D-Hive. A few of them heated up for Simplot, so those are running out the doors because they're not ready to warm up.
Yeah.
it's just a tool to hold them until they can dry up the rot if they do it properly, but if they warm up the bins, it's all over.
Ticking time bomb at that point,
Yeah.
obviously, yeah.
They did everything right, put the big heaters and tricked the system to bring in more fresh air, dry it up, the DMN and the Sorox.
Yeah, so that's a challenge, right? You
Yep.
can't heat it up and dry it out, it's just gonna speed it up.
Once winter came in, a lot of bins, that was it. It's,
Yeah.
We got a storage in Maine that we're working on right now that we're doing a study on it. And we've weighed in two pretty large storages, about 70,000 hundredweight per storage. And they were filled as close as what we could get with similar potatoes. Trucks just showing up, randomly backing up to whatever pile. And we weighed in 40 sample bags in those storages, which these will all be tested at load up. Treated one at door closure, 12.5 parts per million DMN. And at this point, everybody that's walked those piles just obviously notices the difference in the pile that we treated. We went back approximately 4 weeks later and did our CIPC application on both storages same day. But that building that we treated is obviously got something going on, much better storability than the other check storage. And we're 6 months away from getting weight results, disease results, fry out results. But the initial walkthrough, I mean, it's just, you just think you have two different lots of data. You know?
Are
As
they-
far as cost return, will be determined in the spring.
Yeah, yeah, when the rubber hits the road.
Yep.
Are they doing monthly quality samples
What we're
out of there
doing
right
right
now, or
now,
what's
so
going on?
far, is once a month, we're doing the lab tests for DMN residue
Okay.
analysis. And they're doing fry color tests monthly. And, that's it.
Okay.
But the sample bags all came off one truck. So even though they're in different storages, they all grabbed the sample bags. They are all exactly the same potatoes off the same truck.
Okay.
They were going in one of the storages and they just kept throwing them in the other.
What variety were they using?
Russet Burbank.
From Maine?
oh,
What's that?
Maine?
Yep. Yeah, we grow russets too.
That's good. Triers though. Does anyone know the Triers? Work with the Triers?
They're, you know, it's quite interesting.
Yeah.
I think everybody in here is DMN, they love DMN, otherwise they wouldn't be
Yeah,
here.
yeah,
We all know
I'm
what it
preaching to
does.
the choir, right?
Yeah,
That's all.
yeah, a little bit, but it's still a little bit sporadic, Dr. Campbell. It'd be nice to know some information. I really appreciate what you're doing. It's just like, even on frigidity of a potato late in season, we're finding out there's a difference. If the potato was hydrated going into storage and you put DMN on it, it would be, better later. But DMN doesn't help at all a potato that's dehydrated even before it goes in storage. I mean, can you give me some insight there? How that?
You know, the analogy I can give you is that I also work at a grape station. And the grapes go to make wine. And the winemaker says, I just can't make good wine from bad grapes. So the question is, if you've got bad potatoes going into a bin, and you're looking for a magic bullet, it's going to be a tough one, right? I don't know. In a perfect world, the best potato going in the bin is what you want. And that might be the thing where DMN is going to keep the best potato the best potato. If it comes in as something that's really damaged and really just riddled with disease, DMN may be helping, but when the stuff goes to market, it's still going to look like a bunch of diseased potatoes. so I don't know. Yeah.
so in Manitoba, 90% of the potatoes are fresh back as dry land. So 2 years in a row they came in just dehydrated. But part of it is the farmers don't have to sort of compete because it's all quota system, so they're going to sell either way, because they all look bad. All the growers are equal.
Uh-huh.
But everybody, 100% of it's, well, 90% of it's all DMN now. In 2 years in a row, these guys, they just can't believe the difference in their what they're not culling out are bruised, dark spots, like DMN, dehydrated potatoes. Like 2 years in a row, we had 2 inches of rain all summer long on potatoes, and the fresh pack came in bad shape. They're seeing way better results even on bad potatoes.
Now, when you say really bad, they're still taking. There wouldn't be selling in a prime market, right? These potatoes would be competitive.
We have a quota system. People in the
Yeah.
market, they sell anyways.
Right.
There's no there's nobody with better potatoes. They're all equal.
So I think, all right, so DMN is I think is preventing disease, or suppressing disease in the pile. But I think the challenge is that if a farmer has a pile that's really riddled with disease, I don't think anyone should promise them this is going to cure your problem. This is going to help, but it's not going to cure your problem. It's not going away. It's just because bad product is bad product, and you can't turn it into good product. But
So at your level that you're doing in your little dish, your potatoes, you know, the first slide you had out, to me, they look washed. If not, just very clean.
Yeah.
But very clean.
yeah,
So
that was a good year.
would you have the ability at any one of your trials to just take potatoes that say are quite dirty and leave that dirt on them to see what percentage the dirt's responding with the chemical versus some that you'd say dry brushed? So you'd almost have two tests and 2 checks going on at the same time
Right.
to see how much of our product is dirt related, killing the spores that are in the soils, and how much is there in the potato itself. Because typically when we have issues like he's describing, your dirt increases with your rot because your rot's due to moisture.
Right.
So not only are you bringing in more disease, more rot, frost damage, and everything else, you got piles and piles of dirt that don't necessarily come off the potato because it's just so wet.
Well, we have a small side project we did this year. We took some French fingerlings and grew them out at the grape station, which is a good story. And brought them in, treated them with DMN, and put them in storage. But we had tremendous rainfall right around harvest time, and they came in all mud caves, and those two were deep. But we didn't. We're not treating them to do this type of experiment, but I could see your point. Those are really very different than what I saw that Troyer harvested that year. That he's shipping down to Seabed here from Florida for the fresh market. So, yeah. It also depends, I think, on soil type too. That's going to be a big thing on soil type. If you grow in a nice sand, that just falls right off, right? You grow where I am, that's got clods of clay on a each tuber, holds some moisture. I just think in the east we have more problems with that kind of disease storage than I think maybe in the west. Maybe I just think that. You know, grass is always greener on the other side of the Rockies. Well, that's fun. So we're going to continue on this. We're interested in a couple of things that we're going to look at. Also, is what's happening to the pathogens in a little more detail. So what's causing them to slow down in their growth? Because it would be interesting to see if there's a parallel between why potatoes are slowed down in their growth by DMN and what pathogen is slowed down in growth. What is this slowing down in the growth process? It might give us a better idea of what's going on. Earlier studies that we did, we do know that DMN is preventing cell division and shutting down cell division. But that's a very simple kind of story. That's like saying, you know, I turn the light switch and the lights go off. But that doesn't tell you that there's a whole wiring system between the switch and the light, right? And that's essentially what DMN does. You know, I can tell you that DMN induces this gene and that gene does something. Then the question is, well, but there's more to this story than just that single response. It's a lot more complicated. So we'll continue to go on that. And we'll get some better idea, I think, what bacteria do as we go along. the bacterial cultures, it might be something we could put more effort into, and not just work on ones that are interesting to human pathogens, but ones that are interesting to plant pathogens. But then again, you know, I need to stress, you know, it's not that easy as someone who does research to just order a pathogen and have it shipped to you. You have to get little permits to have things moved around. It's easier for me to go buy a MRSA-resistant bacteria and have it shipped to me from a company than it is for me to get a pathogen that infects wheat. If I do that, I have to get all these USDA permits and regulations to be able to have shipped to me. But I get a MRSA strain shipped to me at my home. It's just the strangest world we live in. So it's interesting. But this has been fun. It's real challenging. I love a puzzle, and this is a puzzle to me, and I'll continue to puzzle with it. So any suggestions that you guys have, I'm certainly welcome to your own ideas. Those are certainly nice to know, because you're doing the applied stuff, and you see the stuff in the field, and I don't. I only see the stuff in my little tiny bubble. And that's sometimes bad, because then I forget that there's an actual application to stuff, and there's actually a real world purpose for what I do. Yeah.
So when you talked about the cut potato piece there, so you are doing something right now with?
Yeah, what we did is this is an interesting experiment. We cut the surface of the potato, put one in a control, one in DMN, and then we let them go 2 days, and then what we did is we scraped the surface, and then we isolate bacteria and fungi from the surface. And we then use a technique to sequence, and we just sequence all the species. We don't culture anything. We just figure out something's there because of its DNA presence. So we're looking at the genetic population that sits on the surface of the tuber. What I can tell you is that in 2 days' time at DMN, I didn't see a big difference. So I need to go back to that experiment and figure out, all right, is this because I don't see a big difference in 2 days? There is a big difference between a cut surface and an uncut surface. And both in control and DMN, that if you look at the cut surface of potato, the number of fungi and bacteria on there is really low after 2 days. On an uncut surface, where all the mud and dirt is, it's huge. There's hundreds of fungi and hundreds of bacteria. But when I look at the diversity level, like I count the number of different species I see, I don't see a difference between DMN and control. We didn't set the experiment up right. We need to think of it differently. But I like that experiment. I'm going to do
three years in a row now. We do is cut seed. He cuts the seed and then we DMN it. And now I got 3 growers that they just can't believe the results they're getting now. We're doing their cut seed for dormancy and for what they see in the field for even emergence. And
really? So they see a big
cut
difference?
seed and the whole seed peep at the same time. And
so they cut the seed then treat it with DMN? Or they're treating
starting in
DMN?
January. They're cutting seed right now, all cut. They're going to treat it, put their treatment on there, their dust and their fungicide, and then I'll be DMNing them in about March,
Right.
April. Actually, some we go 15 days before they plant.
So it's like 1/4 seed, and then you're going to then plant out.
Yeah,
in May
they
or
do
whatever.
100%, and I got 2 more growers. They just can't believe the results they're seeing.
That's what I like about DMN, is that you can just go put this stuff back in the ground.
Vigorous plants, they see come out of the ground faster.
Now they're treating with fungicide too, though, right?
Yep.
I don't think I want to. In my experiment. But we're taking the same approach. We're just treating the DMN
Yep.
we cut. And the reason why we did it is it's a perfect comparison, right? Half the tuber goes there, and half the tuber goes there, right? It's an easy, simple approach.
You're
So we'll go back.
treating the cut seed now because you're cutting it now, or?
Nope, they cut seed now. They're filling their bins with all the seed. Keep it warm, superize, then drop the temps, and in about March I'll go treat it with DMN.
So
March, April.
Are you seeing a difference in that versus seed that's treated that isn't cut? That you treated?
They're doing it so they can cut their 6,000 acres worth of seed early, and then they're seeing more even emergence. And then when there's all their other spray applications in the field, and then they see more uniform size at harvest.
But do you treat anybody's potatoes that aren't cutting them now, seed?
Yep.
And you're treating those, seeing the same results?
They're doing it for the dormancy, and these guys are doing it so they can get their cutting done, so it doesn't all sprout in the bin.
But
They're all seeing good results.
When they cut, they're like
been
supervised.
cut and holes. And that's not what.
They're mixed, yeah, like this, all would stay whole, and,
So
So
you're
when
saying
they-
the same as well on
Yeah.
the whole side as well.
yeah.
So when they cut, they're letting it suberize, they're letting it heal
Yep,
up.
they keep more
before
and
they
they
then,
Suberize, I only treat all of them in March.
so they're gonna go like a month or two
Yeah.
after the
I
cut.
see the same thing as he's saying, we just don't cut seed now, but we're definitely seeing big time emergence issues. We have a variety that's special in Maine. It's a very undormed potato, a caribou russet. And they are just, you have to treat them. We're gonna treat them probably twice with 1,4 seed before they plant it.
What, are they out of dormancy now?
Yep.
It's like sick them in reds, and then they cut a sick them in reds.
And maybe in it, some varieties,
Yeah.
just like, huh.
But with that
Mostly-
variety, it'll do an atypical sprout. If you treat it, and I got pictures to back it up, you'll take that potato and every eye is exactly the same.
Yep.
So if you don't treat it, you plant that potato, you're gonna have to make a decision probably in August. Am I gonna kill it to keep these two big 10-ounce potatoes from getting too big to have to cull them and sacrifice the 4 that are undersized? Or am I gonna let these 4 come in and be chuck the 2 big
Okay,
oversized?
so you cut them, and you're treating with DMN. And
I'm
DMN is
cut-
making them, when they sprout, they're sprouting fairly even.
I'm treating them before they cut them.
Right, but you're treating them after they cut them.
Yeah, this-
But
these-
the same
yeah,
thing,
we're doing
you're
the same
seeing
thing.
when they sprout, you're not seeing an apical dominance. You know, the apical end of the
That's correct.
tuber has a fairly strong apical dominance. That bud comes out first, right?
That's
a
correct.
winner. But when you treat with DMN, you don't see that apical dominance.
No, all eyes peak
Yeah.
at the same time. That's what-
That gene that suppresses auxin growth by DMN, think of the Christmas tree. The Christmas tree, the shoot at the top is making auxin. The auxin's coming down and inhibiting the buds. The further away you get from an apical bud, the further away you get from its auxin. And then the more branching you get. So you get big branches at the bottom of the tree, tiny little branches at the top. In the case of the potato, you treat with DMN, you're inhibiting auxin. It says auxin signaling is shut down because the gene for auxin signaling is being screwed up by DMN. That would mean your apical dominance should go away. Huh.
They've done
Excellent.
stand counts
It's
3
good
years.
talking, folks, so now we
3 years
understand what's
running
going
at
on.
At McCain Foods in New Brunswick, we have done the stand studies for 2 years in a row. They haven't given us the data yet, but it's in the 20 to 30% increase in yield because, in this variety,
Right.
because of the stand improvement.
Huh. Wow, that's interesting.
That's marketable yield, not just, you'll still have the same tonnage, but marketable potatoes thereafter. Yeah. That's quite a difference.
Mhm. Thanks for letting me talk.
Any other questions for Dr. Campbell? He'll be here at the end if you guys want to have some private conversations with him too. One thing I do want to point out and add, it's just nice, Dr. Campbell, to see your science begin to match some of the anecdotal evidence we've had from growers and you guys over the years, to see that begin to merge a little bit. It's kind of an exciting time to understand better. I mean, we still got questions like Woz, your question about the hit and miss. Sometimes it doesn't work. We've got some more things to figure out, but to be able to understand, just as we just said there, the auxin and the apical dominance being released, it's nice to be able to see the science behind that as well too. Other thing I wanted to point on is Dr. Campbell's work on the fungistatic properties of DMN. That's important, as 1,4-side is a biocontrol product, and that's a tool we want you guys to be able to go out in the marketplace and use that fungistatic properties. It's not a fungicide that kills the fungus, slows the growth. When its DMN goes away, the growth comes back, and hopefully, like Wazen's question, that's enough that the natural pathogens on the potato will outcompete the Fusarium or some of the other species. That's something we've got to figure out and continue to work on. But as a biocontrol, that's a good selling point to help us go out and do that. So thank you, Dr. Campbell. Appreciate the help there. So Should we take a 5 or 10 minute break, grab some water, use the restroom, and then Addie will be up next. Thank you.